going to be even faster.
Yes.
So, when we think about how cells get to be in tissues and how tissues work together, and also consider that we as people, we start from a single-celled fertilized egg. And this picture is quite nice. It's showing that here is a sea urchin and here is a mouse, and they both start from a cell that looks the same. They both end up with completely different body plans. And sea urchins actually are a very important organism for studying development, believe it or not. There was a lab where I went to graduate school and they studied development and they used sea urchins. So getting from here to here, or here to here even, is a really complex process, and it's important that it go right. It's important that every single cell division on the way is timed correctly, positioned correctly even. That's one cool thing about development, is not only do the cells have to divide, they have to divide off to the correct location. Or sometimes a cell has to crawl from one location to the next during development. So to get to the point of having a good, functioning, complex tissue, like skin, which is not even the most complex tissue, you have to have several different types of cells in their correct layers, in their correct positioning. And so this is showing, if we start deeper down in the skin, the yellow is fat. There are fibers running through that. There are blood vessels running through that. Above you have a lot of connective tissue, which is the dermis, which is also leather. Is that gross to think about? That's what leather is. That shows you how fibrous and tough and strong your dermis is. Then you've got a little bit of intervening tissue, and up here, epidermis. And in all of this, the place that's most cell-packed is the epidermis. So we have cells, they would be stuck together with adherens, junctions, and desmosomes. Down at this level, we would have hemidesmosomes at that border. When you get into connective tissue, the cells are sparse, the fibers are more numerous. And so this would be stretchier and more elastic than this because the fibers are so packed down here. But this is also stronger than this. You've got to have the right cell in the right place, and a mixture of cells. A mixture of cells. So you've got epidermal cells. This is a melanocyte. You also have immune cells in here that squish and migrate through, search for invaders. Everything in the right place, at the right time, with the right neighbors. So depending on what cells are present, what their configuration is, what kind of fibers they have, that will determine the tissue type. And within a tissue, like skin, each different cell in this tissue is supposed to be dividing at the correct rate. Dividing at the correct rate. So epidermal layers are supposed to be having cells divide more frequently than dermal layers. So if you imagine the average epidermal cell in your body, it has a much shorter life, a much faster regeneration cycle than a cell in the dermis or a cell that is in muscle, or definitely a neuron cell. They divide at different rates. And when they divide, cells usually will divide and become the same kind of cell, work in the same way, probably express the same genes, and it will have a certain identity. The cell identity. So the cell identity you can compare to the cell's, its function, its metabolism, its behavior, so to speak, And also its gene expression. What genes is it able to use at that moment? All of that rolls into a cell's identity. Who am I as a cell? Well, it depends on what I hear, so to speak. What do I hear in the environment around me? What do I sense in the environment? What signals do I get from outside? So all the signaling mechanisms that we learn, paracrine, contact dependent, hormonal, and so on. That helps maintain my identity as a cell. Who am I literally stuck with? This is like so people relevant. Who am I stuck with? Who is my neighbor? Who do I spend a lot of time with? What am I bound to? What are my cell-cell connections? And then this one as well. What do I remember about the past? As a person, who I am is based on what I listen to, partly who I'm with all the time, and what do I remember about the past. And those of you that are in genetics, this is the epigenetic stuff. There was no cell memory when I was young. Well, there was. People just didn't know it yet. This is the epigenetic part. What genes do I have locked up tight in chromatin? What histones do I have methylated? Which ones are acetylated? That's what I remember. That relates to what genes can I actually use at this time and which ones are locked too tight. It's a hugely exciting field. But all of that together determines, am I a skin cell? Am I a neuron? Am I a fibroblast? Am I a red blood cell? The combination of those factors determines my identity, and the identity determines what can the cell do? What can that cell do? What job can it do? So if you go back and think about the zygote, what can the zygote do? Well, it can divide. It is not specialized. It's not differentiated. It does not have any mature function. But what it has is possibility to give us eventually every cell in the adult body. It's got possibility. It has no specialization. It's like a baby. Oh, so much possibility. You can be whatever you want when you grow up. Maybe. Maybe. Lots of possibility, not so much function. So that concept of possibility, so I'm writing over here on the side, possibility versus having a specialized function. That's a continuum. And any cell in our body we could put somewhere on that continuum. The embryo, the zygote itself, would be up on this end. It doesn't have, what can it even do right now? Nothing. It has endless possibility, though, to create every other cell in the body. The cells that we call stem cells have lots of possibility, but they don't actually do anything. They're there, they can divide, they've got possibility, they don't really do anything. They're not accomplishing a function because they're not differentiated. So to me, this is like going through educational process. So in some countries, even when you're 7 years old, kids have tests and they're like, okay, you're going to be, you're going into economics. You're going to be a carpenter. You're going to be a teacher. You're going to be a medical person. And then you go through training in just that, just only that, so that you can become that thing. So there's early differentiation into educational tracks. The U.S. system is not quite so rigid. You learn generally for longer and then decide later what you want to do. So those of you who have not decided, you're not totally sure what you want to do, that's a blessing. At least no one told you, you're going to be a skin cell. But the more differentiation, the less potential to become anything. So if I wanted to go back to school now and be, I don't even know, I don't want to do anything else but this, but if I wanted to be an accountant, it would be a little bit more difficult for me to do that now. But I still could. I guess I still could. I'm not going to. So it's important that we have cells in our body that actually do the work and do the jobs and have the function. But we also need to be able to replace them. So we need a mixture of both. And most of our body tissues will have some small number of undifferentiated stem cells somewhere nearby that can give rise to the other tissues. So we have stem cells that stay undifferentiated so that they can keep reproducing and produce the functional cells. This is the lining of the gut, specifically in the large intestine or the colon. And we're going to use this picture for several different things coming up. So the function of this tissue, what is the function of colon lining? Absorption of what?
Water.
It's mostly water at this point. Some nutrients. Like the further down you go, the fewer nutrients are being absorbed. But you do reabsorb water. So the function of this tissue is to, number one, absorb water. What else is it supposed to do? What else is it supposed to do? What's this mucus secreting stuff for? To move things. Yeah. You need to absorb water from the food residues, and once you've absorbed the water from them, get them to move on further down, which mucus helps that process. So you've got absorption, you've got mucus secretion. These cells need to hold together, provide a barrier. They have this shape for better absorption, more absorptive surface. And then down deep, you have some stem cells to help reproduce the rest of the tissue down in these crypts. So that's large intestinal colon lining. This picture is showing a cartoon of skin. So different location, different function. This is external body skin. What's the function of external body skin? What's it supposed to do? Protection. Good. Temperature regulation. Waterproofing is another major one. So this is supposed to be doing something completely different than gut lining, other than cells need to stay together to provide a barrier. So it provides a barrier. We have dead cells on the outside, which can be shed off without really harming us. It makes it difficult for pathogens to get through there. It prevents water loss through the skin. And it also provides for temperature regulations because you have blood vessels underlying down deeper. So in the skin, where do you think the stem cells are for epidermis? They'd be down here, right? They're deeper. That also makes sense because, I didn't hear what you said, but they're deeper. It makes sense because if they're on the outside, they'd be easier to get damaged to. So they're deeper levels. So as cells migrate upward through your skin to the outside, they're gradually dying, but that's normal. So when you look at people and you think that they're beautiful or whatever, you're looking at death. Is that weird? Think about a lot of weird things. Like you're looking at dead cells. When you see people, you're looking at dead cells, except through their eye. That's beautiful. Anyway, you should look people in the eyes. Look at the living part of them. In the blood, so back to earlier, skin epithelial cells, the stem cells for those are down in the deeper layers. Where are the stem cells for blood? Where do they stay? In the bone marrow. Yes. Much more centralized because blood is a circulating tissue. So the stem cells for blood are in the bone marrow, which is why if you have problems with any of these particular cells, a bone marrow transplant can help you. So that type of stem cell curing has been around for a while now. Like, oh, your blood cells messed up or your immune cells messed up. Have a transplant, get some new stem cells for blood into your bone marrow. And that's called hemopoietic or sometimes hematopoietic stem cells. Those are in the bone marrow, gives rise to all of this. So in all of these pictures, you can draw a line from less differentiated, more potential to less potential, more function. Same continuum. So back to the gut lining, down in the lining of the colon. So this is a picture of that same tissue we saw. Here's the crypt. This is the crypt down here. The stem cells are right at the bottom of that. There's the stem cells down in the crypt. And they divide, and as the cells of the epithelium divide, they sort of, in effect, push upward. The lining of the colon is renewed from the base. So they push upward. Cells can get sloughed off up here, but they get replaced from the depths. And this process is mostly controlled by this protein called Wnt. It's W-N-T. You say it Wnt. So this is talking about the Wnt pathway, Wnt signaling pathway. And Wnt is a diffusible, we need to write this down, this is important. Wnt, W-N-T, is a diffusible, floating, short distance signal. It's a signal molecule. So Wnt is made down low. It does not go very far. This would be like a paracrine signaling. Short distance. It's not going into the bloodstream. It's acting locally down here. And Wnt tells these cells in the tan to divide. It's a mitogen. It's a signal to divide. I don't think I've talked about mitogen yet. Let's put that on here. So Wnt is a diffusible short distance signal, so it's paracrine signaling. And it is also, in addition to that, it is also a mitogen. As in mitosis. Mitogens. Mitogens encourage more mitosis. What I just wrote is, and also a mitogen, which is a division signal. So a mitogen is some kind of chemical or protein or substance that when you provide it to cells, they start doing division. It's an old word. We don't use it much anymore, but you might see it. Mitogen for mitosis promoter. So wherever Wnt can float to, cells will divide. It's a signal. Go ahead and divide. It seems like I'm totally switching topics. Later on, after Thanksgiving, we talk about a lot more detail of Wnt signaling. I just wanted to set it up here, so that's why I'm suddenly not talking about it anymore. We will later. So what I want to have here right at the end is set up a little bit for the presentations we're going to hear this afternoon. And what they're generally about is, I want to go back to this picture. Can we take a cell that is medium along this pathway, not totally differentiated, not totally undifferentiated, and use it to make it go backward? Can we make it unlearn what it knows and then become a precursor to some other kind of tissue? We have like a few different talks about this. So, like, can we take a cell that is partway down the skin pathway and make it turn into a neuron? I don't know if that's the real example. Can we take a cell that is partway down the, what is some other, is everybody starting with fibroblast? Okay. It's usually a fibroblast. What do yours start with? Astrocytes.
Thank you.
Astrocytes. Can we take an astrocyte, which is a neural precursor cell, and make it what? What's it going to turn into?
Neuron.
Into a neuron. Yeah.
That actually makes sense.
Can we take something out of the middle of this pathway and make it go backward, in essence, so that it can produce a different kind of cell type? Because when people have a problem with their tissues and they need a new version of it for some reason, I need a new version of muscle, I need a new version of neurons, mine are messed up, I need a new version, nobody needs a new version of fat cells, but I need a new tissue because mine is broken, so to speak. There are ways to do this. So when I was your age, the talk was, we could totally do this if we can use embryos because they have all the undifferentiated cells. They could become anything. And the question was, well, should we use stem cells from embryos to try and start to help cure Parkinson's? And that's a tough question. That's a tough question. So the debate back then was, should the government even fund any kind of research on this so that we could maybe make replacement healthy tissues for people with disease? And at that time, I'm not going to go through this actually because I know you talked about this in genetics, but no. We'll go to here. So at that time, it seemed like, well, if we don't study these embryos, that's our only hope. That's so 1990s now. And I can't tell you how nice it is to hear these presentations of these papers, to take a medium stem cell, not totally undifferentiated, take a medium skin cell from a skin culture that's not killing anyone. I mean, that's a loaded statement on its own. Dispensable, let's say. And do some things to it to make it go backwards. This is the process of going backwards. This is a stem cell for skin, a fibroblast. We're knowing more now about how to undo the epigenetics, how to turn on regulatory genes to make this undifferentiate, go back, become more flexible, so that then maybe we can grow new tissues to help people's disease without using embryos. That's fantastic. That's fantastic. And doing this couldn't happen until we knew more about epigenetics, until we knew more about promoters, which might seem boring, but they're not. Some of you are probably going to talk about transcription factors, critical for this process. So we'll be hearing more about this afternoon in presentations, but We're going back in time. Can we make new tissues? From your papers, it seems like that's well on its way to happening. So remember to be here at 1:10. We've got the order set up. I'll have your score sheets. We'll be ready to go. Thank you.